Okay, so now we're up to muscular system part 3, and we're going to be looking at muscle contraction. So the 3 learning objectives that we're going to cover today include explain how the muscle fibers are stimulated, the events of excitation, contraction, coupling that lead to the cross-bridge activity, and then the sliding filament theory. Now, the reason we're looking at all 3 together is because you can't really have the last one, number 6, without 4 and 5. So by now you should be quite familiar with an action potential. So we need to have an action potential or an electrical current that will help to excite the nerves and then consequently here the muscles. We have something called excitation contraction coupling, and this is the series of events that links that electrical signal and turns it into a mechanical action or contraction. We also have to have motor neurons. These are nerve cells that will activate the skeletal muscle. And of course, we have axons, which are the extensions of the motor neurons leading down into a synapse. So we need to discuss a motor unit, and a motor unit is a motor neuron plus any muscle fibers that it innervates. So it's important to note that one motor neuron could innervate more than one muscle fiber. Groups of motor units often work together as a motor pool to coordinate the contraction of a single muscle. When a motor unit is activated, all of its fibers contract. So this is an all or none phenomenon. The force of a muscle contraction is controlled by the number of activated motor units, but we will return to this concept later. So in order for a muscle to contract, it must be firstly stimulated by a nerve ending. It must then propagate that electrical current or action potential along the sarcolemma and into the muscle fiber. And we must also have a rise in intracellular calcium, which triggers that final go sign for contraction. So we actually break down how a muscle contracts into 2 parts. So after we've passed the message from the brain to the muscle, we then must link that electrical signal to the contraction or to the mechanical action, and we refer to this process as excitation contraction coupling. Part 2 is the actual contracting of the muscle or of the sarcomere, and this uses a process called cross-bridge cycling. So part 1 of this process is the excitation contraction coupling, and this involves 2 phases, each with a couple of steps. The first phase is activation. Step 1 here is that the fibers must be activated, in that they must be stimulated by a nerve ending so that we have a change in the membrane potential. Step 2 is it must be sufficient to generate an action potential in the muscle sarcolemma. Phase 2 is the true excitation contraction coupling. Step 3 is when the action potential is automatically propagated along the sarcolemma deep into the muscle tissue. And step 4, we have that rise in intracellular calcium levels to ensure that final trigger for contraction occurs. So phase 1, step 1, involves a structure called the neuromuscular junction. Typically, as we mentioned before, they are situated midway along the length of a muscle fibre. The neuromuscular junction is formed from the axonal endings, which have small membranous sacs or synaptic vesicles that contain the neurotransmitter acetylcholine. It also consists of a motor end plate of a muscle. This is a specific part of the sarcolemma that contains acetylcholine receptors. Although exceedingly close, the axonal endings and the muscle fibers are always separated by a space called the synaptic cleft. So this is a flowchart to describe what happens when the action potential arrives at the axonal terminal. So the first step is for the action potential arrives at the neuromuscular junction, and this causes the activation and opening of voltage-gated calcium channels. We therefore then have an influx of calcium into our axon terminal. This calcium increase then signals the neurotransmitter field vesicles or the synaptic vesicles to move towards the synapse. The vesicles then bind to docking proteins and release the neurotransmitter into the synaptic cleft via exocytosis. The neurotransmitter will then diffuse across the synaptic cleft and binds to receptors on the postsynaptic membrane. This will then initiate a signal along the sarcolemma of the muscle. So if we look at it in a picture, we can see here that the action potential arrives as it comes down the axon. It causes the voltage regulated calcium channels to open and calcium enters the axon terminal. It then causes the synaptic vesicles filled with neurotransmitter to bind to a docking protein and then release those neurotransmitters to the synaptic cleft where they can bind postsynaptically and then elicit an action potential along the sarcolemma. But remember, there's always going to be a slight delay as we are going from an electrical signal to a chemical signal and then back to an electrical signal. But it's also important to note that we don't want our muscles to be stimulated all the time, so therefore we need to break down that acetylcholine. It is most commonly degraded by an enzyme called acetylcholine esterase. Remember that if you see A's at the end of a word, it typically means that it is an enzyme. Acetylcholine esterase breaks down acetylcholine into choline and acetate. The choline is then transported back into the axon terminal and recycled to make more ACH. Interestingly, inhibitors of the enzyme prolong the life of acetylcholine, and there are certain nerve gases and organophosphates that lead to an accumulation of acetylcholine and its associated toxicity, causing continued muscle contraction. Conversely, acetylcholine receptor agonists are used to treat diseases such as myasthenia gravis, which is an autoimmune disease that causes muscle weakness, So we can use a continued contraction to assist people with certain diseases. So we've now looked at phase 1 of the muscle fibre stimulation. So we've had the action potential arrive at the axon terminal at the neuromuscular junction. We release the binding of ACH to the receptors. We have a change in the postsynaptic membrane permeability. We have depolarization of that membrane and the beginning of an action potential in the sarcolemma. We now need to move on to phase 2. So as an overview, we are going to have the action potential travel across or along the sarcolemma. The action potential will then travel along the T tubules. We'll release calcium, and we have binding of calcium to the troponin and the myosin, exposing the myosin binding sites. And then, of course, we'll have the cross bridge formation. So that's part 2, the binding of myosin heads to actin and the beginning of contraction. So the first step is that the action potential is propagated along the sarcolemma and, more importantly, down into the T tubules. This then causes the voltage-sensitive proteins in the T tubules, which cause the calcium channels in the sarcoplasmic reticulum to open. Calcium ions are then released from the sarcoplasmic reticulum and flood the muscle fibre. It's also important to note that the calcium is actually stored in the terminal cisterna of the sarcoplasmic reticulum and released from there. So looking at the full picture, we've already discussed the action potential arriving, ACH being transferred across the synaptic cleft, propagation of the action potential along the sarcolemma down into the T tubules, changes in protein, opening of calcium channels, and the release of calcium into the muscle. This causes troponin to change shape, removing the tropomyosin from the binding site and allowing a contraction to occur. This point we will come back to in just a moment. However, after the contraction has occurred, we remove the calcium via active transport and we actually put it back into the sarcoplasmic reticulum to be used again for future contractions. So the contraction cycle involves a series of interrelated steps. It begins with the arrival of calcium ions within the zone of overlap between the actin and the myosin in a sarcomere. The calcium ions bind to troponin, weakening the bond between actin and the troponin-tropomyosin complex. The troponin molecule then changes shape, rolling the tropomyosin molecule away from the active sites on actin and allowing interaction with the energized myosin heads. Once the active sites are exposed, the energized myosin heads bind to them, forming cross bridges. So once the active site on actin has been exposed because of the troponin binding to calcium, the myosin head is now able to attach and forms a cross bridge with actin. It then undergoes what is called a working or a power stroke. The myosin head pivots and pulls the actin filament towards the center of the sarcomere. In doing so, adenosine diphosphate and a single phosphate are released. The myosin cross bridges will then alternate between attaching and detaching as it moves down, pulling the actin filament closer, very much like a ratchet action. To detach, a new molecule of ATP will attach to the myosin head, causing the cross bridge to unform and returning the myosin head into what is known as the cocked position, which is a high energy state. So another way of looking at that detachment is via this little flowchart. So the myosin head binds the actin when the tropomyosin moves out of the way. The ATP's activity of the myosin head causes the release of phosphate and energy. This results in a structural or a conformational change in the myosin head, which pivots and pulls the actin via that power stroke. This pulls the thin filament towards the center of the sarcomere. At the same time, we release the ADP from the myosin. New ATP will bind, detaching it from actin, and we hydrolyze the ATP into ADP and phosphate, which remain attached to the myosin, and is now in a new cocked position, ready to attach further along down the actin. So what we've been talking about thus far is the sliding filament theory. It is the generation of force. It does not necessarily cause shortening of the fibre, but what happens, it's when the thin filaments slide past the thick filaments. Shortening will only occur when the tension generated by the cross bridges on the thin filaments exceeds the forces opposing the shortening. So we have two words. We have contraction, which is the activation of the myosin cross bridges, and relaxation, where the cross bridges become inactive and the tension that has previously been generated declines. So the thin filaments slide past the thick ones, which means that there has to be some degree of overlap between the two filaments in order for this process to occur. As I said before, it's like a ratchet action, where the bonds are broken and reformed several times throughout a contraction. Think of it as pulling on a rope as part of a team effort in tug of war. This sliding occurs simultaneously in sarcomeres throughout the cell. Things to note as the thin filament moves towards the center of the sarcomere, that the distance between successive Z discs or lines is reduced. The I band will shorten. The H zone, which previously consisted of just thick filaments, will disappear and can only be identified when the muscle is relaxed. And whilst the A bands do not change in length, they are closer together between subsequent sarcomeres. So now we have taken that electrical impulse from our nerve and had it result in a muscle contraction. So we started by taking that action potential, releasing neurotransmitters across the synapse, binding postsynaptically, and eliciting an action potential that travels along the sarcolemma and through the T-tubules. We then have that flooding of calcium from the sarcoplasmic reticulum or the terminal cisterna that binds to troponin, altering its shape, pulling tropomyosin out of the way, exposing the actin binding sites. We can then form an actin-myosin cross bridge Using ADP, ATP, we have a power stroke or multiple power strokes and pulls the thin filament across the thick filaments, shortening the sarcomere. And if we have sufficient tension produced, the muscle will shorten. Remember to rest. ATP causes the myosin head to detach. The troponin, tropomyosin, will block the binding because the calcium has been taken back up into the sarcoplasmic reticulum.